Hello everyone. In this session, we will be discussing the structure of cell membrane. Okay. So first, brief history of study on plasma membrane structure. The first insight into the chemical nature of the out boundary layer of cell were obtained by Overton of the University of Zurich during the 1890s. Overton knew that non-polar solute dissolve more readily in non-polar solvent than in polar solvents, and that polar solute had the opposite solubility. So next is the permeability test. So in this to test the permeability of the outer boundaries layer, Overton placed plant root. Okay, this is plant root into the hundreds of different solution containing diverse areas of solute. Okay, and then he discovered that the more lipid soluble the solute, the more rapidly it would be enter into the uh, plant root cells. Okay, and then he concluded that the dissolving power of the outer boundaries layer of the cell mesh state of the fatty oil. That means that fatty oil are easily dissolved into the lipid. Okay, so next is the insight of lipid bilayer. So the first proposal that cellular membrane might contain lipid bilayer was made in 1925 by two Dutch scientists, first Gorter and second Greenland. So what they done in their research? Uh, these researchers extracted the lipid from human red blood cells, okay, RBC, human red blood cells, and are measured the amount of surface. This is okay. The surface area the lipid would cover when spread over the surface of water. Gorter and Greenland. Okay, they conclude that the plasma membrane contain a bimolecular layer of lipid, that is the lipid bilayer. Okay, so next is the lipid compositions of membrane. So the ratio of lipid to protein in membrane varies for depend on the first it is the organism, the type of organism like bacterium, plant and animal. Now second is the type of cell, which sorts of cell are for example cartilage, muscles and liver. Now next is the cellular membrane, for example plasma membrane, either endoplasmic membrane or Golgi apparatus. Okay. So this is the lipid to protein, it varies. Now the for instance the inner mitochondrial membrane, okay, has very high protein ratio. This is the protein. Okay, this is the protein and its lipid. So the protein ratio is high compared to the blood cell plasma membrane. It's 52, and myelin sheath it's 30. Where is the lipid in mitochondrial membrane is less 20. Okay, blood plasma it is high 40, and myelin sheath is high 80. So this is the structure of myelin sheath. Okay, so in structure of myelin sheath, the concentric layer of plasma membrane that have extremely low protein and high lipid. Okay, so low protein 30 and high lipid is 80. So the particularly lipids are present in myelin sheath. There is a glycolipid called the galactosaccharide. We will discuss in next slide. Okay. So the basic structure of lipid. So this is a bilayer of lipid structure. Here is the hydrophilic head, okay, and hydrophobic tails. So now the lipid outer side is extracellular side and cytoplasmic side. Okay, so two leaflets of the bilayer contain different types of lipids. Is show indicating the color with the sky and pink. Okay. So next is the type of membrane lipids. So first glycerolipids, phospholipids and sphingolipids. Whereas the phospholipids and glycolipids are polar lipids, and storage lipid, that is a neutral, that is the triacyl glycerol. Okay. So several forms of triglyceride and phospholipids found in the cell. So the leftmost scheme is triglyceride, okay, formed by 3 carbon. Okay, this glycerol is composed of 3 carbons, backbone with fatty acid tail bound to the each carbon. Okay, this is second leftmost scheme is a phospholipid bearing 2 fatty acid chain with the third carbon. Okay, a glycerol bound to the phosphate group. So the middle scheme is an ethyl glycerolipid who shares with the two previous structures the 3 carbon glycerol backbone and is slightly modified with the ether group. This is the ether group. In second right scheme, the sphingosine. Okay, sphingosine is shown as backbone instead of glycerol. Okay, this is the sphingosine instead of glycerol. Okay, the sphingosine is shown backbone instead of glycerol, and first fatty acid tail of glycerol is modified slightly. Okay. This is sphingo phospholipid since it bears phosphate group. It's called this sphingomyelin. Now, rightmost scheme shows the sphingolipids called the glycolipids. Since the phosphate group is replaced, okay, with the sugar. So the sugar, there is carbohydrate groups, that is the sphingolipids. Okay, this is glycolipids. Okay. So this is the structure of glycerol, 3 carbon hydroxide group, 3 fatty acid chain, and they form the triglyceride. Okay, so most of the membrane contains the wide diversity of lipids. Okay, and all are lipids which are amphipathic, that is they contain both hydrophilic and hydrophobic ratio. There are three main types of membrane lipids. First is phosphoglycerides. Okay, second is sphingolipids, which non-glyceride lipids, that is not contain the glycerol backbone, and cholesterol. Okay, so first we discuss the phosphoglycerides. So most membrane lipid contain a phosphate group, which is makes them phospholipids. Okay, this is a phosphate group. So because most membrane phospholipids are built on glycerol backbone, okay, this is a glycerol, they call the phosphoglycerides. Unlike the triglyceride, okay, so triglyceride which have 3 fatty acid are not amphipathic membrane, and always the membrane glycerides are diglyceride form. In diglyceride, only 2 of the hydroxyl groups of the glycerol esterified to fatty acid, right? So 2 is the hydroxyl group of the glycerol are esterified to the fatty acid, either saturated fatty acid or unsaturated fatty acid. Okay. And third, okay, so third is esterified to hydrophilic phosphate group. So here is the hydrophilic phosphate group, and this is the hydrophobic tail. The phosphate, just phosphate and 2 fatty acyl chain, for example, 1 and second, and that's just phosphate. The molecule is phosphoric acid, and which is virtually absent in most membrane. Okay, so membrane phosphoglycerides have additional group linked to the phosphate. So glycerophospholipid first is the for example, if phosphate it binds to the, okay, so phosphate group, that choline, okay, forming the choline. Okay, then if the instead membrane phosphoglycerides have an additional group linked to the phosphate, So most commonly either choline, okay, or ethanol amine, next is serine and inositol. Each of these group is small and hydrophilic and together with the negatively charged phosphate, to which it is attached to form the highly water soluble domains. Okay, so this first is diallyl phosphatic acid. This is the phosphatic acid. Okay, phosphate binds diphosphatyl glycerol cardiolipin, that is a 2-phosphate glycerol backbone. Next is phosphatic acid, only hydrogen. Okay, 2 fatty acid chain, phosphate and hydrogen. Phosphatyl choline, that choline compound binds to the phosphate head. Phosphatyl serine compound binds to the phosphate, and ethyl amine, that binds to the phosphate group, and inositol, that binds to the phosphate group. Some of the lipid composition in biological membranes. Okay. Human erythrocyte, human myelin, beef heart, mitochondria, E. coli. Okay, so now these sphingolipids. Okay, so sphingolipids, it is the less abundant class of membrane uh, called the sphingolipids. Are derivatives of sphingosine and amine alcohol. Okay, sphingosine and amine alcohol that contains a long hydrocarbon chain. Here is the sphingosine structure. Okay, that is the NH3, okay, amine alcohol, and that is the sphingosine is nitrogen containing amine alcohol. They also are amphipathic. Okay, so this is a sphingosine is also amphipathic. They having a polar head group and 2 nonpolar fatty acid chain. They also are structural components of cellular membrane. So now the sphingolipids consist of sphingosine. Okay, so sphingosine, first is sphingosine, and they link to fatty acid chain. Here is the fatty acid chain R. It's linked to the its amino group, and this molecule is called the ceramide. Okay, so these sphingolipids are divided into two subcategories of sphingomyelin first, and second is sphingolipid. So the sphingomyelins are the only class of sphingolipids that are also phospholipids. So the general structure of sphingomyelin is here. You can see here the same. It is like a sphingomyelin. Okay, so sphingomyelin, that means the group they present, they attached to the carbon chain. This sphingomyelin. Now glycosphingolipids or glycolipids are built on the backbone structure of ceramide, which is amine derivatives of sphingosine. That is types are cerebrosides, sulfatides, and gangliosides. So first is cerebrosides are characterized by the single monosaccharide head group. Okay, so this is the ceramide structure, ceramide residues, and they bind the carbohydrate residues, okay, monosaccharides, and they form the cerebrosides. Okay, this is the carbohydrate residue glucose, ceramide residues, and they bind and they form the cerebrosides. Next is sulfadides, okay, sulfate group containing derivatives and binds to the cerebrosides. Gangliosides are glycolipids possessing oligosaccharide group, okay, more than 2, that means 2 to 10, okay, 10 monosaccharide chain, including one or more molecules of N-acetylenic neurotransmanic acid. Means they contain the one or more molecules of sialic acid. That is gangliosides. Okay. Sphingolipids, okay, they form sphingosine plus fatty acid ceramine plus phosphoride choline, sphingomyelin, ceramine plus simple sugar, cerebroside, ceramine plus cluster of sugar, gangliosides. Okay, so third group of lipid, it is the cholesterol. So the cholesterol that is mostly present in the animal cell. Okay, they constitute up to 50% of the lipid molecules in the plasma membrane. But plant cell contain cholesterol like steroid. Okay. However, biologists or scientists disagree as to whether they complete lack of cholesterol. So in this figure, you see here cholesterol molecules shown in green colors, okay, of lipid bilayer are oriented with a small hydrophilic end facing the external surface of the bilayer, and the bulk of their, okay, this is the bulk of their structure packed among the fatty acid tails of the phospholipid in there. The placement of cholesterol molecules interferes with the flexibility of the lipid hydrocarbons chain, which tend to stiffen the bilayer while maintaining its oral fluidity. Okay, so this is the all about the today's session. I hope this will help you. Thank you.